Okay. Assalamu alaikum. Am I audible? Please let me know.
Yes, doctor. Walaikum assalam.
Okay. So we are doing basic science classes. So first we are doing the past medicine topics reading with conception, then solving the few questions from the past medicine. So last classes we have done until the cardiac action potential, as it is very important in both in the cardiology, also in the basic science. So again, I am just showing the picture like this is the action potential. So the depolarization means action potential. So when the depolarization is happening in the cardiac myocytes, when potassium is in flux, so potassium inside, then depolarization happened. After that, when potassium influx happened, then if you're seeing the screen, you're seeing that this potassium and sodium is entering in the cell. That means there is rapid depolarization happened in the cardiac myocyte. And after that, potassium is out from the cell, also some chloride, and that is transient out. That is early phase of the repolarization. And after that, if you see the plateau phase, this is plateau phase. This is due to calcium ion in, due to negativity inside the cell. Then calcium cell is entering inside the cell. Due to potassium is out, then it is sharp fall to the resting phase. Then another action potential is happening from this part. So action potential have depolarization, then starting of the repolarization, there is some phase, then again starting of another depolarization, that is the cardiac action potential, what we are seeing in this picture. So what kind of ion is entering inside the cell and why this has happened? In this box, if you see, there is phases 0, 1, 2, 3, 4. So very importantly, all the phases are very important for your exam. So mostly they are asking all the phases. So 0 phase, that's mean rapid depolarization, rapid sodium influx. These channels automatically deactivate after a few milliseconds. So that's mean rapid depolarization in the phase 0. Sodium entering inside the cell. Then after that, 1, that means early repolarization. After depolarization, there is repolarization. This is early repolarization. So after sodium influx, there will be efflux of the potassium from the cell. Sodium entering into the cell, and after that, at the starting point of the repolarization, potassium is going out from the cell. Then the plateau phase, slow influx of the calcium inside the cell. And at the phase 3, final repolarization, that is efflux of the potassium from the cell. And 4, restoration of the ionic concentration. So after restoration of the ionic concentration inside the cell and the outside the cell, another depolarization is starting to happen. So that's mean it is a repeating process. So we need to know the phase 0 to 4, in which phase what is happening. So early repolarization, plateau phase, final repolarization, and restoration of the ionic concentration, how it is maintained and which type of ion is responsible for which type of phase. That is important for exam. So anyone can take a screenshot of this box and share it in the group. By this you will can memorize later on. Okay, so next topic is cardiac physiology. So from here, left ventricular ejection fraction. So as we have done with the cardiology, Dr. Rubab, if you are here, can you see the screen? Then let me know which investigation is very important to detect the ejection fraction of the heart. Ejection fraction of the heart. Another one is seeing the arterial lumen, whole of the cardiac structure. Cardiac structure, whole of the cardiac structure. How we are seeing that. Another one, lumen of the vessel. lumen of the vessel. So it is very important.
Doctor, for this EF go for echo, for cardiac structure will go for movement, and for lumen will go for angiography.
Okay. So ejection fraction, we can detect the ejection fraction by doing the echocardiography, ejection echocardiography. You are right, echocardiography, but mostly in the exam they are asking about the Muga scan. Muga scan is most specifically using for detecting the ejection fraction. And whole cardiac structure we are seeing by MRI, cardiac MRI. We are seeing by cardiac MRI. We are seeing the whole cardiac structure by using the cardiac MRI. And lumen of the vessel, that is coronary arterial lumen, we are seeing by using the angiography. So that's mean angiography. That is why we are using the percutaneous coronary intervention as a treatment of ischemic heart disease like stable angina, unstable angina, and angiography is a investigation of choice to detect any arterial luminal obstruction by atheromatous plaque or something. So lumen we are seeing by the angiography. So angiography, we are detecting the coronary arterial lumen. Angiography. Another one, cardiac structure, we are seeing by using the cardiac MRI. And ejection fraction, we can see by using both echocardiography. Another one is Muga scan. Yes, Dr. Ruba,
But
would you
doctor,
like to say?
you were saying that EF usually, but very rarely we are doing Muga scan for ejection fraction.
Yes, ejection fraction, we are seeing the Muga scan.
What do you think? Okay, okay. So ejection fraction, if in the exam they are going to ask, then we are going to mark it as Muga scan, not as echocardiography. And for cardiac structure MRI and for lumen angiography, we know.
See, are you seeing the screen now?
Yeah, I'm seeing.
So Muga scan, multi-gated acquisition scan, also known as radionucleotide angiography, and maybe perform as a stress test. So these things are not asked in the exam, but sometimes they're asking what type of nucleotide is using. That is technetium 99M. This type of radionucleotide is using in this test. That is technetium 99. Sometimes they're asking. So if you see nuclear imaging technetium 99, this is mostly asking in the exam for basic science. Another one for that is position emission tomography. What kind of dye is using? That is fluorodeoxyglucose. And if you see the past paper of previous dyes, then you can get this kind of question like what kind of nucleotide is using and what kind of dye is using in case of PET scan or CT scan like that. So that is fluorodeoxyglucose is the answer. Another one is technetium 99 is the answer. And for Muga, also technetium 99 is using. And C, can accurately measure left ventricular ejection fraction. So this is mostly asked in the exam. Which investigation can accurately measure the left ventricular ejection fraction? That is Muga scan. Is that okay?
Okay, doctor.
Okay, another one. Also keep in your mind. So from Muga scan, you need to know only the dye. So nucleotide. Another one, the last line is important. And if you see the final portion of this line, say typically used before and after cardiotoxic drugs are used. But just keep in your mind, sometimes they are giving some more detail in the question, then you can use it as a key. Another thing, keep in your mind that is accurately measurement of the ejection fraction, that is Muga scan. Okay. Now the left ventricular ejection fraction. So some physiology we have to know. See, left ventricular ejection fraction, how you will measure, that is stroke volume divided by in diastolic volume into 100. So these calculations sometimes they are asking, as it is basic science, so you have to know. So mostly they are asking, that is left ventricular ejection fraction, how you will calculate that. Another one is stroke volume, how you will measure, that is in diastolic volume, in diastolic volume of the left ventricle, and in systolic volume of the left ventricle. By this, you can calculate the stroke volume, and after calculating stroke volume, you can put it in this calculation, and you can calculate the left ventricular ejection fraction. This is physiological method. Another one, cardiac output. Cardiac output is stroke volume into heart rate. So stroke volume and heart rate. So you need to know how you will calculate the stroke volume. And pulse pressure, that is systolic pressure minus diastolic pressure, that is pulse pressure, and factors which increase pulse pressure. A less compliant aorta, this tend to occur with advancing age. So advancing age, so some geriatric people or very elderly age, they have this kind of thing. Another one, increase stroke volume. And systemic vascular resistance, how you will measure systemic vascular resistance, mid arterial pressure by dividing it by cardiac output. So from this topic, you need to know how to calculate left ventricular ejection fraction and stroke volume. Another one, factors increase pulse pressure. Factor increase pulse pressure. So increasing age, or advancing age is a cause of the less compliant aorta. Another one, increased stroke volume. These two factors increase the pulse pressure. Now cell cycle. So cell cycle, you need to know the phase and in which phase what is happening. So G0, that means resting phase, quiescent cells such as hepatocyte and more permanently resting cells such as neurons. So this is resting phase, quiescent cells and the neurons. So the quiescent cells, that means this kind of cell are not usually entering into the mitosis or cell division. So these cells is in resting phase. And G1 phase, so those cell who is going continuous multiplication in our body, this kind of cell is entering in the G1 phase, that is gap 1. In this gap 1, cell size increase and determine length of the cell cycle. So G1 is the gap 1. So as it is a gap phase, so this will increase sometimes, and this is determining the cell cycle length under influence of the P53. So this G1 phase is very, very important, because we know under influence of the P53, this phase is happening. And also gap means if the gap is increasing, that means cell cycle the length of the cell cycle will increase in this phase. And P53 is very important because mutation in the P53 genes, this happen, this condition takes place in some individual, and that is a condition where there is lots of cancer happen in those individual. That another the most common name mostly asked in the MRCP is, that is Lee-Fraumeni syndrome. Lee-Fraumeni syndrome mostly asked in the exam, Lee-Fraumeni syndrome. So I'm sorry, my asthma is aggravating nowadays. So, okay. So G1 phase, P53 is responsible for this phase, because under influence of the P53, G1 phase is happening, and this is determining the length of the cell cycle. And here cell increase in size, And now S phase means this is, so G for gap, Z0, that means 0. So it is resting phase. These cells are in resting phase in this phase. And S means synthetic phase. Synthetic phase
Sorry to
where,
interrupt.
yes.
You mentioned in G1 that there is a syndrome. Can you please repeat it?
Well, yes, we will repeat that. I will show the syndrome. No problem. After
Okay.
finishing this one.
Okay, thank you.
So
Thank you.
S phase, synthesis of the RNA. S phase means that is synthetic phase. So synthesis of the DNA, RNA, histone. This is important for the new cell from the mother cell. So sorry, the mother cell and the daughter cell, we need to know that is there is some synthesis of the protein. So that is important. So synthesis happen in the S phase. Okay. Now Z2, Z2, that is another gap 2. Cell continue to increase size. So G means gap 1, gap 2, and in between gap 1, gap 2, there is S. So first gap 1, there is increase in cell size. S, as cell size is increased, S means there is some synthesis of the protein. And in gap 2, there will, after synthesis of the protein, increase the cell size more until the requirement. Then M phase, that is mitosis, cell division, the shortest phase of the cell cycle. So M phase means the cell increase in size, cell have adequate protein, and our cell got its maximum capacity to divide. And in the M phase, that is mitotic phase, so cell is prepared for division. So so cell division happen in the M phase. So mitosis is M phase, that is cell division happen here, the shortest phase of the cell cycle. Keep in your mind, mitosis, that is M phase, is shortest phase of the cell cycle. If the shorted phase is increasing in our body, that means there is some carcinoma. That means there is some carcinoma. Because we know the oncogene, so our cancer cell has uncontrolled cell division and multiplication happen in maximum capacity. That means there is M phase is high or lengthened, so cancer cell are increasing in size and forming a tumor. So it is Allah gifted, God gifted thing, mitosis, cell division, the shortest phase of cycle. In normal individual, it is shortest phase of cell cycle. So all the things you have to read in a focused way, and then you need to learn how this cell cycle is happening. Dr.
Doctor, I
Ruba,
just
would you like to summarize?
sorry.
Yeah.
Sorry to bother you. Understood that G0 is the resting phase. G1 cell size is going to increase and it is determine the cell cycle. Okay, and then S phase, there will be synthesis. So it means S phase will be synthesis of DNA, RNA, and histone and centrosome will be duplicated. And G2 will be cells will continue to increase. And as you mentioned, that is the shortest of the all the cell cycles. And if it is increased, then there will be cancer. Okay, in the body. And G1 phase, you also told that it is somehow associated with Leigh-Fraumeni syndrome, P53 also. So can you explain
P50.
that?
Yes. I'm discussing that. P53. So just memorize this box now because we are reading the cell cycle. So all of these things are important, and I added few things that is in G1 phase, that is under influence of the P53, this G1 phase is happening. So if any mutation is happening in the P53 gene, that is a condition raised in one's individual's body, that is Leigh-Fraumeni syndrome. It is mostly asked in the MRCP. It is very important. I am going to discuss this now. Another one, that is M phase, and it is God gifted. See, the shortest phase of the cell cycle. So mitosis, what we need until that extent, mitosis is happening. But if the mitosis length is increasing in our body, the length is increasing, that means these cells are going to form a tumor that is abnormal cell in our body, and that is obviously abnormal. So this is God gifted. Mitosis is the shortest phase of cell cycle. Okay, so all of you are asking the Leigh-Fraumeni syndrome, so let's discuss it. Hello. So, okay, I am trying to search it in past medicine, but I am not getting it. Maybe it's under. Okay. Anyway, so Lee-Fraumeni syndrome, it is a hereditary and genetic disorder that increase the risk of some carcinoma in a family. So all the family member have that risk. So are you seeing the screen or not? I'm not getting you because there is network problem.
Yeah. We are seeing the screen. No issue. It's clear.
Okay. So yes. So that is Lee-Fraumeni syndrome. So Lee-Fraumeni syndrome is a condition. Lee-Fraumeni syndrome. It is a condition. It is arise in an individual that has hereditary predisposition of developing carcinoma in a family. Okay, so it is happen in a family when there is mutation in the P53 gene. P53 gene. So Lee-Fraumeni syndrome is a condition that is a hereditary condition. That means there is mutation in the P53 gene, and in this condition there is chance of developing some carcinoma. What kind of carcinoma? That is breast carcinoma. So this family member have this kind of risks of developing breast carcinoma. Another one, soft tissue sarcoma. Soft tissue sarcoma. This is obviously in past medicine, but I don't know why I am giving the search, but it's not coming up. But obviously, if you have the online past medicine, if you try to search it out, it will get the topic. And I will share it later on. That is Lee-Fraumeni syndrome, breast carcinoma, soft tissue sarcoma. Another one is leukemia. And in some extent, there is chance of developing
And
adrenal carcinoma.
Dr. Jasmine, sorry,
Yes.
Dr. Jasmine, sorry to bother you. You search for, you write only P53, it will come.
Okay.
Write only P53, it will come.
It's not coming in my past medicine. Why? Not coming up. Yeah, now it's so CP53 is a tumor suppressor gene located on the chromosome 17p. It is the most commonly mutated gene in breast, colon, and lung cancer. So if you the writing are present in the screen, right? So see, they mention about the breast carcinoma, they mention about the colon carcinoma and lung carcinoma. So another one that is sometimes they're giving the sarcoma. So soft tissue sarcoma. Sarcoma means connective tissue disorder. Sarcoma also some sort of some people in the family member there is a history of blood cancer, that is leukemia and adrenal carcinoma. So all of these things are related with this. So P53 is the thought to play a crucial role in the cell cycle, preventing entry into the S phase until DNA has been checked and repaired. So in S phase, DNA should be, so in S phase, those synthesis of the DNA, RNA, and some protein, or what is required for the new cell, that is synthesis happen in the S phase. So before entering the S phase, these P53 have a role to check the DNA whether it is safe or not. That is checked and repaired. If there is any problem, that is repaired. So if the problem within the P53 gene, that means there is chance of developing this carcinoma. And Lee-Fraumeni syndrome is a rare autosomal dominant disorder characterized by the early or set of a variety of the cancers such as sarcoma, breast cancer, leukemias. So you can put in you like a number: 1, breast carcinoma; 2, sarcoma; 3, blood leukemia, blood cancer; and 4, lung cancer, colon cancer. So let's clean it. So from the top to bottom, if you so 1, so from the chest, see breast carcinoma; 2, lung carcinoma; 3, there may be chance of developing and in the abdomen, colon carcinoma; 4, you can say that is also in the abdomen, colon carcinoma and adrenal gland carcinoma sometimes; 4, leukemia; and A is connective tissue disorder, that is sarcoma. So 1, 2, 3, 4, 5, 6. So 6 type of carcinoma can happen, and this has history of different family member have different type of carcinomas, as it is autosomal dominant condition. Again, keep in your mind, it is hereditary condition and it is autosomal dominant condition. Yes.
Dr. Jasmine, can I ask you one question? Is the video recording for everyone or just for your enrolled students?
Only for enrolled students.
Okay, thanks.
Welcome. Now cell division, and from cell division, and it's from cell division you can read the whole topic for your own learning, but for exam I am focusing only for exam. So for exam you need to know the phases, and for if you see that during the S phase of the cell cycle, the cell prepare itself to division by duplicating the chromosome. The table below shows the phases of the mitosis itself. So the phases of the mitosis is important, and in prophase, chromatin in the nucleus condensed. So phase are prophase, prometaphase, metaphase, anaphase, telophase, and the cytokinesis. So this phase is important, and as I shared in the group, I am preparing some notes. So with some mnemonics, I will share it eventually. So in prophase, chromatin in the nucleus condense. Prometaphase, nuclear membrane breaks down, allowing the microtubule to attach to the chromosomes. Metaphase, chromosomes aligned at middle of the cell. And at the middle of the cell, and after entering in the middle of the cell, that it is going to divide. Anaphase, the paired chromosomes separate at the kinetochores. So here, cell aligned in the middle of the cell, and anaphase, here the paired chromosome start to separate, and in telophase, that's mean chromatid arrive at opposite pole of the cell. Telophase, so it is like you can memorize like tunnel. So it is pulling all the chromatin to the peripheral part of that cell, and in the middle it is starting to divide. And cytokinesis, actin myosin complex in the center of the cell contracts, resulting it is being pinched into the 2 daughter cells. So this is the picture. So I will copy and paste this picture in the WhatsApp group after the session. Now cell organelle. So cell organelle. So past test has lots of question from this, only from this box. You will get lots of question in the past test from cell organelle. But you need to know the very few things for those question, what is mostly asked in the MRCP. So endoplasmic reticulum. So there are 2 types we know, that is rough endoplasmic reticulum. Another one is smooth endoplasmic reticulum. So rough endoplasmic reticulum, translocation and folded of new protein, manufacture of lysosome enzymes, and site of ink-link glycosylation. Example of cells with extensive rough endoplasmic reticulum include pancreatic cell, goblet cell, plasma cell. So sometimes they are asking which kind of cells are cells have very extensive amount of rough endoplasmic reticulum. So if you see here, pancreatic cell, goblet cell, and plasma cell. So just keep in your mind, that is pancreatic cell, goblet cell, and plasma cell, they are going to secrete some hormone or some enzymes or some kind of things in our body. So that needs more rough endoplasmic reticulum, because rough endoplasmic reticulum is related with the processing of the protein, and you see translocation foldation of the protein enzyme, like that. So our pancreatic cells have secrete lots of hormones like insulin, somatostatin, gastrin. So lots of hormones are coming from our pancreatic cells. And also goblet cell, they are secreting some mucus in our body, because this is also one kind of protein. Another one, plasma cells. Plasma cell going to secrete some antibodies, and plasma cells are all the antibodies are derived from the plasma cells. So these cells are related with secretion of the enzymes, secretion of the proteins, secretion of the hormones. So these cells require more rough endoplasmic reticulum in their cytoplasm. So this is mostly asked in the exam. And smooth endoplasmic reticulum, they are also steroid and lipid synthesis. So here is mostly, we know the enzyme, hormone, and some functioning protein. Rough endoplasmic is related to the protein, rough protein, RP. Rough endoplasmic reticulum is related with the protein synthesis, RP. Rough with the protein, and smooth is related with the steroids or lipid. Smooth. So see, example of cells with extensive smooth endoplasmic reticulum include those of the all the structure, organ, or cells which have huge amount of protein. They are related with the protein, fat metabolism. See, adrenal cortex, it is fatty area. Hepatocyte, we know all the hepatocyte, it is fatty area. Testis is also fatty area, and ovaries is a fatty area. This needs hormone. So this is coming from this is usually in steroid in nature. So these cells are mostly related with the smooth endoplasmic reticulum. So these cells have plenty of the smooth endoplasmic reticulum. So you need to know this one, you need to know this one. So smooth endoplasmic reticulum is responsible for steroid or lipid, and rough endoplasmic reticulum is related with the protein. So those cells who have more involvement in the protein processing or protein secretion, processing, production, those have more rough endoplasmic reticulum, and those who have relation with the steroid hormones, and they are consisting extensive amount of the smooth endoplasmic reticulum. As Dr. Rubab is there, would you like to review this topic, because very important this one? Dr. Rubab or Dr. Shormila, anyone unmute yourself and read this two line, only this one and this one. Then I am proceeding. I proceeded to next question. Okay, I think everyone is busy. So next topic, that is Golgi apparatus. See, modifies, sorts, and packages these molecules that are destined for cell secretion. The addition of the mannose-6-phosphate to protein designated transport to the lysosome. So as you already seeing in the screen, that is mannose-6-phosphate to protein designated transport to the lysosome. This is important. So Golgi apparatus, sometimes they're asking this one. See, from Golgi apparatus, you need to know only this one, that is this addition of the mannose-6-phosphate to protein designated transport to the lysosome. So Golgi apparatus have relation with this one. And mitochondria, that is related with our aerobic respiration, contain mitochondrial genome as a circular DNA. And nucleus, we know the function of the nucleus, that is DNA maintenance, RNA transcription, RNA splicing. And lysosome is breakdown of large molecules such as protein and polysaccharide to the smaller molecules in our body. nucleolus, that is ribosome production, and ribosome is related with the translation of the RNA protein, and paroxysm, that is catabolism of very long chain fatty acid into amino acid. So from all of these things, the endoplasmic reticulum, function of the endoplasmic reticulum is very important. After that, you need to know the relation with the Golgi apparatus. That is, this one is very important from this box. And the function of the paroxysm and proteasome. Sometimes you will get confused which one is related with which. So paroxysm, catabolism of very paroxysm. So it will catabolize the large fatty chain, fatty chains and amino acid. So fat is lipid, amino acid is protein. So it will catabolize the long chain fatty acid and amino acid and result in the formation of the hydrogen peroxide. And proteasome, so it is protein, proteasome. So it is paroxysm, so peroxide. Proteasome, along with the lysosome pathway involved in degradation of the protein molecule that have been tagged with the ubiquitin. So the clue is ubiquitin. When you will get the ubiquitin in the question option, then it is related to the proteasome. And proteasome is related with the lysosome. In the name is same, lysosome. Proteasome is some sort of, they are acting in same manner. And when you will get ubiquitin in the question or in the scenario, ubiquitin means it is proteasome. And paroxysm, peroxide, paroxysm, that is result in formation of the hydrogen peroxide, and they are related with the catabolism of the long chain fatty acid and the amino acid. So these are the most frequently asked topic in the MRCP exam, and you will get lots of question in the past test from the cell organelle. It's very important.